Hello, I'm Jeanne Dutron, a student in neurosciences. In this video, I'm going to talk about a fascinating study published in PLOS Biology by Bruckner and colleagues. Can the gut microbiota, the microbes that live in our digestive system, influence how social behavior develops? You may have heard of this before, that the gut microbiota plays a major role in digestive processes in nutrient absorption. But guess what? That's not all, and you're going to discover all of this with me through this video. The aim of this video is to explain to you how the microbiota communicates with the brain through the immune system, and more precisely through microglia. Let's define what microglia are. They are the resident immune cells of the brain. They play a key role during development because they sculpt brain circuits by eliminating unnecessary neuronal connections. To illustrate this point, think of microglia like gardener trimming a tree. They cut away the extra or misoriented branches so that the tree can grow in a healthy shape. In the same way, microglia remove unnecessary neuronal connections so that brain circuits function properly. The thing is that many psychiatric and developmental disorders, such as autism, are characterized both by altered social behavior and by immune system dysfunction. Understanding how microbes shape the brain could help us find new ways of thinking about these conditions. The main objective of the study was therefore to test whether the microbiota influences social behavior by acting through microglia in the forebrain of the zebrafish. Why the zebrafish? Because it's a powerful model organism, because their larvae are transparent, allowing neurons to be observed and immune cells to be searched for directly in the living brain. To test their hypothesis, the authors compared three groups of zebrafish larvae. The first group was germ-free, which means it was raised without any microbes. The second group was conventional, with a normal microbiota from the beginning. And the third group was called ex-germ-free. It was initially raised without microbes, but the researchers introduced bacteria on the seventh day. The scientists then measured three different aspects. They observed social behavior by testing how much time a fish spends oriented toward another fish behind a transparent barrier. They studied the morphology of specific neurons in the forebrain. They also analyzed the microglia by counting them and studying which genes they express thanks to a technique called single-cell RNA sequencing. You must know that this combination of methods is very powerful because it links three levels of analysis: behavior, brain circuits, and immune cells. In other words, it allows the researcher to show not only correlations but also possible causal relationships. So, what did they find? Let's have a look at this now. First, behavior. Germ-free fish showed reduced social behavior. They spent about 30 to 40% less time facing another fish compared to controls with a normal microbiota. Next, neurons. The number of neurons was similar between the groups, but germ-free fish had branches that were much longer and misoriented. What this shows is that without microbes, neurons fail to form the right connection. Finally, regarding microglia, germ-free fish had fewer microglial cells in the forebrain. Importantly, when the researcher artificially eliminated microglia in normal fish, these animals also showed altered social behavior. This basically means that microglia are necessary for the development of normal social interaction. Finally, the authors performed rescue experiments. When they reintroduced certain bacterial species into germ-free fish, they were able to restore normal neuronal morphology and normal social behavior. What is striking is that not all microbes are equal, but some are sufficient to promote brain and behavioral development. But then, what conclusion can we draw from all this? In conclusion, the importance of these results is that they establish a causal chain. The gut microbiota influences microglia, which remodel neuronal connections in the forebrain, and this remodeling is necessary for normal social behavior to emerge. However, there are several limits to consider. The zebrafish is a non-mammalian model, so the results need to be tested in mammals before we can generalize. Also, the gemfish condition is artificial, since in real life most animals are colonized by a diverse microbiota at birth. Finally, even though the study identifies complement signaling as one mechanism, many other signaling molecules are probably involved and they remain to be explored. I like to round up by saying that this work demonstrates that the microbiota is not only important for the digestion and immunity, but also for the brain wiring and social interaction. Paves the way for research into disorders such as ASD in mammals. One final question this raises is which precise microglial metabolites send signals to microglia? This could be a lead for future research. Thank you for watching.